The central dogma of biology. DNA is the instructor, which makes RNA, which is the messenger, which makes protein, which does all the work.
So the DNA has a double helix, and it was discovered by Watson and Crick, um, and they say they were explaining the secret of life.
DNA scripts. T goes with A, C goes with G. Inside the DNA, which is inside the nucleus.
So there are four nucleotides: the thymine, the guanine, which are pyramides, and the cytosine and the adenine, which are purines.
Okay. One of the strands of the DNA is the sense strand, and the other one is the antisense strand. Both together makes the DNA double helix.
The cell is conformed by a nucleus. The nucleus contains the DNA, which, as I already said, converts to RNA, which is found in the cytoplasm. And the DNA to your left is an example of the steps that are shown. The first step is the division by the helicus, which is that small circle.
The second step is that the primase starts replication and makes main stand. Here, the primase start replications, so one strand goes to the new cell and the other one stays with the old cell.
So in the step 3, the DNA polymerase completes the antisense strand or
The sense strand in
Yeah.
other cases.
yeah. So at the end of all, the DNA ligase seals the Okazaki fragments to finish the replication process.
Next page.
Transcription. Here we see the RNA make a version of the DNA, which is why we put the RNA versus DNA. It shows the two difference between the two. RNA has a single-stranded cell. DNA has a double strand. RNA uses ACGU, while DNA uses ACGT. So instead of being ACCGT, it would be ACGGU.
So the steps of the transcription are this. Step 1, the helicase divides the DNA just as in the replication. But this time, in the step 2, the RNA polymerase gets one strand and changes it into an RNA. It changes the script.
In the step 3, RNA is out of the nucleus and is now in the cytoplasm.
Translation. In this one, you can see the messenger RNA bringing in groups of 3 of the ACGU. Here, the ribosome comes and makes these groups of 3, converts them into proteins.
So in the step 1, the RNA letters are divided in groups of 3 to form protein and amino acids. Then step 2, the ribosome slides through the RNA and makes them separate proteins.
And in step 3, the proteins end up being folded proteins as seen in the upper part of the page, like this one. Thanks.
That's all. Thank you.
Subscribe for more.